Second talk is made, in fact, by Alice Vilela. So Alice is a professor in UTAD, University of Trás-Montes and Alto Douro. Alice, in fact, received the Bachelor of Science degree in Enology. She has a master degree in genetics and PhD degree in microbiology. Currently, she is a professor at the University of Trás-Montes and Alto Douro. She is member of Chemistry Research Center of Villarreal. Her research lines are linked with a study on volatile acidity bioreduction in wine and food and wine sensory evaluation. She operates in the area of food science with an emphasis in microbiology and sensory analysis. She is also coordinator for the UTAD testing panel that was formally constituted in 2010. And the panel participates in several research projects and support several wine and food related industry in Douro region. Alice will present to us biomodulating wine acidity, the role of non-Saccharomyces yeast. It's a very important issue, as all of us know that, of course, the acidity can become a problem with climate change. And so it should be interesting to see the presentation of Alice. And Alice will be able to answer your question as she is here. Thank you, Julien.
Okay, my name is Alice. And first, I would like to thank Professor Pierre-Louis Tessèbre and Fernando Zamora for the kind invitation for this meeting. And I'm going to talk about the biomalation of wine acidity and the roles of non-Saccharomyces yeasts. So, in recent years, in line with consumer preferences and because of global climate changes, new trends have emerged in wine fermentation and wine technology. Consumers are looking for wines with less ethanol and more fruitier aromas, but also with a good balance in terms of acidity and mouthfeel. Non-conventional yeasts contain a wide range of different genera, and they are also important for wine modulation. In the past, they were considered spoilage, but now they are used to enhance the aroma profile of wines or to modulate wine consumption. Recent publications highlight the role of non-Saccharomyces as selected strains for controlling fermentation, mostly in co-fermentation with Saccharomyces. In this presentation, I have reviewed the ability of some bacteria and also non-Saccharomyces strains to modulate wine's acidity. So, organic acids and sugars are the most abundant solids present in grape juice. They are significant constituents of juice and wine, and they are responsible for sour and acid taste. They also influence wine stability, color, pH, and other important qualities. The quality and quantity of grape organic acids in conjunction with the sugars has a significant effect on the mouthfeel of wines. So acid composition and concentration within the grape mass or wine are influenced by many factors, such as grape variety, soil composition, and climatic conditions. During the winemaking process, it is advisable to monitor the concentration of organic acids in order to ensure the quality of the wine. And a distinction is made between the acids directly produced in grapes, like, as you can see in the table, tartaric, malic, and citric acids, and those originated during the fermentation, like succinic, acetic, and lactic acids. So these acids can be seen in the table and their associated aroma or taste that modulates really the wine's pleasantness. I'm going to talk a little about acetic acid because it's considered a spoiled characteristic in wines. So in quantities higher than 0.8 or even 0.9, it presents a vinegar smell and a very acrid taste, causing the wine to be considered spoiled. The maximum acceptable limit for volatile acidity is 1.2. It is a huge amount already. Acetic acid can appear on the grapes or in the grape mass due to the presence of yeasts like non-Saccharomyces yeasts, or even filamentous fungi like aspergillus, and bacteria like lactobacillic-like bacteria and acetic acid bacteria. If the amounts are higher, some contamination, spoilage, yeasts, and bacteria can also be present, especially at the end, like Candida, Pichia, and Sectobacter bacteria. After malolactic fermentation, heterofermentative species of Oenococcus oeni and Lactobacillus also have the potential to produce acetic acid due their possibility of metabolize the residual sugars present in the wine. The wine biological acidity modulation by bacteria via malolactic fermentation can be done, while during alcoholic fermentation, wine yeast strains convert the grape sugars into ethanol and other compounds. After sugar depletion and the decline of its population, lab will proliferate by using the remaining sugars and thereafter perform malolactic fermentation. Of course, the metabolism of Oenococcus oeni can improve the wine's sensory characteristics because it produces secondary metabolites. However, malolactic fermentation can be influenced by very enological parameters like temperature, pH, and SO2 content, and so on, and also its inhibitory metabolites. So several works are now focusing on alternative bacteria, such as Lactobacillus plantarum, that you can see an image, beautiful drawing image in the slide. And this lactobacilli can perform malolactic fermentation in wine. They can survive under winemaking stress conditions, and during the fermentation process, they are also able to produce a huge number of secondary metabolites. And very interestingly, they can improve wine's color and solve problems associated with filtration due to tannase activity. Moreover, Lactobacillus plantarum are considered probiotic, so they improve human health. So they are beneficial to wine and also to the wine consumer. In this table, we can see the percentage of the ethanol formed during fermentation and sugars fermented, main volatile compounds formed, and effect on wine acidity of 7 non-Saccharomyces yeasts. From the list presented, only Schizosaccharomyces pombe, Lancia thermotolerant, and Toleraspra delburgi are presently produced in industrial level by biotechnological companies, so they can be bought to perform this biological modulation of wine's acidity. The first one that I'm going to talk about is Lancia thermotolerance, because I worked with a strain of this yeast in my PhD degree a few years ago. This yeast is known to be able to perform wine acidification as also the acetification. So one worldwide problem is the increased alcohol content with the concomitant reduction in the total acidity of wines associated with global climate changes. Since 2013, studies have been made in several wines and wine regions that elucidate Lancia thermotolerance winemaking features. This yeast can increase lactic acid concentration and reduces pH. However, Lancia thermotolerance is also able to do another very interesting thing, that is, it can be used to develop a controlled biological deacidification process, so it can use acetic acid and decrease high levels of volatile acidity, with the process being oxygen dependent, which means that its metabolism must shift towards respiration than fermentation. And here we can see in this Petri dish the first time that I isolated one Lancia thermotolerance. It was coded 44C in my lab, and we have made further studies with it, and it really could have a good decrease in the acetic acid content once it decreases the acetic acid from 1.2 grams per liter to 0.06, almost zero, in a biological deacidification process. And I know that there is already a commercial strain, but is only sold for its potential for the lactic acid production due to the sugar's consumption. So further studies are needed to be done, because I see a great potential in this yeast also for the acidification process. Okay, another interesting is Saccharomyces pombe. It can perform biological acidity modulation via malloalcoholic fermentation. It's highly appreciated in colder regions due to its metabolism of malloalcoholic fermentation, and it can significantly reduce the levels of ethyl carbamate precursor and biogenic amines that are compounds that have very hazard health not beneficial, so they can perform health issues. It increases the formation of vitisins and other pigments that intensify the colors of the finished wines, and there is already a commercial strain available also in the market that can be bought and used to perform malloalcoholic fermentation. Another interesting is Torollaspora delbrueckii. It can modulate wine acidity in very sweet wine. So Torollaspora delbrueckii was studied in 1981 in order to evaluate the possibility of using it to improve the quality of botrytized wine. These wines are made with grapes with a very high sugar content and present a challenge to Saccharomyces cerevisiae. In terms of acetic acid production, it is well known that cerevisiae, when is a very osmotic stress present of a very high sugar content, it produces high levels of acetic acid as a stress response to that high osmolarity. So the volatile acidity of these wines may be over 1.8 grams per liter. That is very high, and really above the legal limit, that is 1.5, and 1.5 it is really high. So some authors have used mixed cultures of Torollaspora delbrueckii and Saccharomyces cerevisiae in high sugar fermentations with a higher concentration of delbrueckii. So they were able to promote its growth, and this mixed inoculum produced lower levels of acetic acid and acetaldehyde. That is also a very dangerous product and not a very pleasant one in wines. An indication of the positive impact of Torollaspora delbrueckii activity in wine quality was also demonstrated in 2015 in one work performed in Vino Santo, a sweet wine, due to its low production of acetic acids. So I know that there is already a commercial strain available that can be used to perform these fermentations and use in very sweet musts. Now I'm going to talk a little bit about Candida Stellata. This strain is not available in the market, but we know that it can do biological acidity enrichment. And why? Because it produces succinic acid. So during the beginning of alcoholic fermentation, many species can grow simultaneously in the grape must, and Candida Stellata has been described in this stage of fermentation. An interesting feature in the ability of Candida Stellata is the ability of this yeast to form succinic acid. The higher production of the acid was also associated with higher glycerol content, so the final wine was very mellow, very soft, and this also presented high ethanol production and ethanol tolerance. However, succinic acid could positively influence the sensory mouthfeel profile of wines with insufficient acidity, but due to its salt, bitter, and sometimes sour or excessive sour tastes, a huge level of this acid would negatively influence wine's quality. I think we must try it more in laboratory studies and then see how well it performs the acidity enrichment, and if really the final mouthfeel it's not salty or it's not too bitter, and we only feel the freshness of the acid. Zygosaccharomyces florentinos, also called Zygotoraspora florentina, is also an important yeast that can modulate volatile acidity of wines. Here we have another important yeast for decreasing the acetic acid content. It can be used in mixed cultures with Saccharomyces yeasts. It was used in a very promising study with Lalvan yeast, and increased the production of propanol, higher alcohols, and esters. So the final wine was really very interesting and very fruity, and the acetic acid levels were very low. The reduction of volatile acidity and improvement of polysaccharides and 2-phenylethanols was also fine. So a rose-like aroma, along with the fruity character, was also found. And at the winery scale, with red grape mass from the Sangiovese grape variety, it's an important work that I've read. The final wines presented a higher concentration of glycerol and esters. So sensorially, the wine's astringency was lower, and the aromatic character was well defined. And well, have a very pleasant feature at the end. Another interesting yeast is Pichia kudryadzevi. It can perform also malic acid consumption, so it could be used also as Zygosaccharomyces pombe. However, besides being able to do this interesting malic acid consumption and production of significant levels of glycerol, and besides the final wines present fruity and cookie pears aroma, it was also able to reduce the production of ethyl acetate and produce very pleasant esters, very fruity esters. However, as I said at the beginning, it can cause some hazards in the health of the consumer, because studies were made very recently in 2018, and it was shown that Pichia kudryadzevi is very related in terms of DNA, so they are almost like sisters, with 99.62% identical in DNA sequence with Candida krusei. And we know that Candida krusei is responsible for about 2% of its infections caused by Candida in humans, so it will not be safe for now until further studies are needed to see if we can really use this strain to perform the malic acid consumption in wines. Okay, one interesting yeast that must also be studied further is Starmerella bacillaris, also known as Candida zemplinina. It can perform biological acidification and deacidification. Okay, so malic acid degradation due to its ability to produce a wide spectrum of extracellular hydrolytic enzymes. It can consume malic acid. It is also capable of producing pyruvic acid, acting as a natural acidification agent by reducing the wine's pH. And the use of this yeast in mixed fermentations with Saccharomyces cerevisiae could be beneficial and reduce acetic acid also in sweet wines, leading to a decrease of 0.2 grams per liter of acetic acid. And it can also reduce ethyl acetate. And as we all know, ethyl acetate, it is an ester, but it has not a fruity aroma. It has a glue-like aroma, so it is not pleasant to smell in wines. In Sauvignon Blanc wines, the fermented by these mixed cultures contained significantly higher levels of esters and thiols and were considered very pleasant in terms of aromatic characteristics. So, for finalizing my presentation, I have here some conclusions that I think they are important. Many non-Saccharomyces yeasts present interesting enological properties in terms of fermentation, purity, and production of metabolites or even ethanol. When used alone or in mixed culture with Saccharomyces cerevisiae, these yeast strains can modulate wine acidity and increase production of some interesting compounds that can give the wines fruity aromas and flowery aromas. They are really very pleasant. Non-Saccharomyces yeasts that are most often considered spoilage now can be seen as a tool for the winemaking industry. Of course, they must be studied, mainly those that are not yet commercialized, but maybe in the future, if they are studied more, we can use them. A deeper study of the enological traits of these yeasts will provide new data for consideration in the control of fermentation, with special regards to warm climate wine and botrytized sweet wines, where they are commonly found in mixed populations. So I want to thank you all for your attention, and I hope you be safe in this time, in these dark times of pandemic, and also drink wine and be happy.
Hi, hello, Professor.
Hello. Thank you, Alice. It was very interesting talk. Alice, before to go to the question of the audience, I have maybe 2 or 3 questions for you.
Of course.
So your talk was really very interesting because of the situation with especially acidity, of course, but also there is also secondary compounds of interest, especially for the flavor of the wine. First point, we know that there is volatile acidity and fixed acidity. So do you believe that some of the species that you were talking about, or maybe some other, have more interest for the fixed acidity or for the volatile acidity?
Well, for my opinion, and for my experience in my work, I think more for the acidification, for the removing the volatile acidity, I think.
Okay. Another point is concerning natural yeast, because I think that all of these species that you show or strain are natural, probably. So
Yes.
Yes. So the point is to know what they can target in terms of acidity. Is it one acid in particular for some of them, or is it several acids, in fact?
Okay. For example, Lancia, we know that it produces lactic acid, but can also decreases the volatile acidity. So I see here a double work of the same yeast strain, and that it's very important. For the other, for example, Torulaspora, okay, Torulaspora is interesting, but mainly used in sweet wines because it is more, it can tolerate more the sugar concentration, and thus the fermentation, she will not produce a higher concentration of acetic acid as it should produce, it would be produced by the Saccharomyces cerevisiae. So here, I think what it is important is to know what the problem is, what is our grapes, and what are we going to do with them? What is the purpose of our wine? If we want a fresher wine, we must, and we have high sugar grapes, we must use a strain. If we think we don't have problems in acetic acid, so we don't use it. We use only the Saccharomyces cerevisiae. I think who makes the wine must know very well and what is the raw material, how are the grapes. For instance, acetic acid can be present at the beginning of the fermentation. It can be present in the grape mass, so it will be a huge problem, because if we vinify grapes that have already 0.3, 0.4 grams per liter of acetic acid, we will certainly end with a very high volatile acidity. So taking all into account, I think an enologist that is informed, that goes to the field and really looks to the grapes, and then he knows what he must do to make a better wine.
Okay, thank you. This is interesting as answer, because there is in fact finally the first beginning of the fermentation, the first time of the fermentation, and then the end of the fermentation. So there is several aspects, in fact, to consider there. And so maybe my last one, and then we will take the question of the audience.
Yes.
In your opinion, should we go for genetically modified strain to better control acidity in the wine?
Okay, there is a genetically modified organisms. They present a very, I think, a problem in global way, because the way that they manipulate the DNA maybe can promote some problems for our health. And then maybe not, because now we have also CRISPR technique, and CRISPR is a technique that is safer. I wrote a paper about it, and because it is a very interesting point. But I think we need to explore more the world, all that is around, and driven our studies to the natural things. And what is natural? We have a huge biodiversity everywhere in our grapes, or all over the world, and I think we must explore that first, because it is more natural.
Yes, I fully agree. We have to use biodiversity. Thank you for your answer.
Okay.
So we can take some of the assistants. Manuela Brando is asking, we try non-Saccharomyces yeast to prevent acetic acid production during pre-fermentation cold maceration instead of during sulfur dioxide. Do you think it is worth it?
Well, I don't know. Maybe some of them, because I think most of them probably will not work. It's a very tricky question, because I've not worked with it. I'm just thinking about the consequences, because most of them also may produce acetic acid. That is the balance that we must try here. Even Lancia, that I use in some of my studies, when I use it directly in some musts, and I haven't used any SO2. Also, I have pasteurized the must. In some studies, she also produces acetic acid. Also, of course, she was also able to consume some of it, but she also produced it. So I don't know. Maybe not. I cannot answer to with a specific yeast. I think it is a problem. It is interesting, but must be studied to try to see what non-Saccharomyces is able to perform that.
Okay. Okay, thank you. So another question is from Maya Dalla Valle. Have you done any study with sequential inoculation of various non-Saccharomyces yeast combined with Saccharomyces yeast and impact on wine acidity, flavor profile on Finnish wine?
Okay, I have not done it, but I have read some interesting works where they were very successful and the volatile acidity was reduced and the flavor profile was enhanced because, as we know, non-Saccharomyces, they have a huge beta-glucosidase activity. So normally they can really enhance the profile of wines, namely the floral notes of the wines. So I think it is interesting. Sorry, Professor, I can't hear you.
It's okay, it's okay.
Okay, now
Sorry.
I hear you.
Another one is from Maria Cazo. She is asking which non-Saccharomyces you will recommend to decrease significantly the pH of a must using as a bioprotection?
As a bioprotection, what does Maria Cazo means with that? What bioprotection? How? Sorry, I didn't understood the question.
Yeah, I think that bioprotection means that you are using a strain, in fact, that can generate, for example, a compound to protect, in fact, the product. Some of the strain, for example, can generate, I don't know, sulfur dioxide or something else.
Oh, I don't know. Maybe to decrease pH, I think Saccharomyces bombi, well, doing malo-alcoholic fermentation, it will decrease it, and maybe Torulaspora. I don't know. Maybe try that, or try those strains, because I say these two because these two they are already studied. Okay, so the other ones that I've mentioned, there are not a lot of studies of them because we have Steramella bacillarius, for instance, that can decrease malic acid, but there are few studies. It's very new in the literature, the works that have been published. So there are works from 2015. It's very recent. We must, I think, study harder to see. Do you have any other?
Sorry, I am saying that there is plenty of other questions.
Okay.
Is asking you, do we have something more about the key working condition of the use of Lancia thermotolerance to reduce the volatile acidity?
Okay, that's a very interesting question. It can be used, of course. You must take into account that it doesn't work as well as in terms of producing the wine as Saccharomyces cerevisiae. I think it would be interesting to use it in sequential fermentation. So I think it is. And I think it will work better if you perform the biodeacidification in a way that is not very normal. It's not a winemaking process, a normal winemaking process. Is if you have a spoiled wine, for instance, from the last year, and in this year you are at making the new wine. If you take one third of the wine that is spoiled, that has an high volatile acidity, even 2.5 for volatile acidity, and then put that wine with 2/3 of a newly made must and ferment all together, I think that Lancia will take the acetic acid in a very interesting process and metabolize it. And why? Because you will dilute the acidity, so it will not be the 2.5 grams per liter. Of course, it will be diluted to 1/3, but 1/3 it will be very high, and Lancia can consume the acetic acid of the mixture and perform the rest of the fermentation.
Okay, we have maybe, thank you. We have maybe just the time to do two last quick one. One is very interesting and congratulations from Georgios Kosteridis.
Yes, hello, Georgios.
Is there any sensory analysis results?
Yes, in my studies, I have made some sensory analysis. There is one point that is not very positive, is the formation of a little bit of acetaldehyde, okay? And as we know, acetaldehyde, not only in terms of smell, it has not a very agreeable, but this is an hedonic question. There are some people that like smell of acetaldehyde, but it can cause some health-related issues, you know, because acetaldehyde, it can cause headaches and so on. So I think that is the major problem.
Okay, thank you. And the last one for this session, did you work on Metschnikovia pulcherina? Any effect on acidity from
No,
turbot?
no, with Metschnikovia, no. It's a very beautiful yeast, and my students, they love it because in the Petri dishes it has a very beautiful color. But no, I have not worked it in the fermentations. I have my contact with Metschnikovia was in a work where we have made a selection of the yeast strains that were present in the grape mast at the beginning of the fermentation, and yes, we found it there. It was there, but then as the fermentation occurred, she was the population of Metschnikovia decreased it as of all the non-Saccharomyces yeasts, and of course Saccharomyces dominated the fermentation.
Okay, so thank you very much, Alice. Great talk, and you had a lot of questions.
Yeah, and Professor, if I'm able to talk just a little bit more, I have an article published in IVES review that can be read because it is free, and I've also published an article in Fermentation that is also a free journal about this, and of course I presented my email at the beginning of the speech, and everyone that has a question can also send me an email. I am available to other questions that I have not answered here. Okay,
thank you very much, Alice. It was very interesting and passionate, so we were happy to have you and to have you talk.